Hello, thank you for joining us today to discuss some exciting data with Context Therapeutics. Management's prepared remarks will be followed by a brief Q&A. Please welcome our presenters, Eric Butz, Scientific Lead, Dr. Joe Rucker, Research Lead, and Martin Lehr, Chief Executive Officer. If you have any questions during the presentation, please submit them through the Q&A tab at the bottom of your Zoom panel. We'll do our best to address as many questions as time allows.
Really appreciate everyone joining today. The team is currently live in Orlando, Florida at the American Association for Cancer Research annual meeting. For those of you unfamiliar with AACR, it is the largest early stage oncology meeting, meaning preclinical through typically phase 1 clinical trials, where the latest, newest groundbreaking research is presented. And this morning at the event, we had a poster presentation with our most recent preclinical data for our lead program, CTIM-76, our bispecific antibody targeting Claudin-6 and CD3. So before we begin, I would encourage everyone to review our disclosure statement and any forward-looking statements that we may make during this presentation. So I thought it'd be helpful just to spend a minute talking about CTIM-76, what it is. CTIM-76 is a bispecific antibody. The structure is presented here on the left portion of the slide. For those of you unfamiliar with bispecific antibodies, they are antibodies which are proteins that our bodies make to address and fight pathogens. And in this case, a typical antibody is monoclonal, meaning it binds to the same protein on both arms of the antibody. And the arms would be the portions in the top right and left of the Y-looking like structure. In our case, we modified a typical antibody and converted it to a bispecific, meaning it binds to two different proteins. In this case, we wanted to therapeutically exploit the concept of immunotherapy. So with one arm of the antibody, we bind to Claudin-6, which is enriched in many different types of tumors, as well as a second arm that activates CD3, which is expressed on immune cells. And the idea is to bring immune cells in close proximity to the cancer cells to get our own immune systems to essentially attack the tumor. There are some really interesting attributes of this program, particularly our hopes that it will have a wide therapeutic window. That would be the difference between the efficacious dose that would have an impact on the tumor and tumor biology versus unwanted safety. And so our hope is that we can generate very significant efficacy before we would see unwanted side effects. And there are some structural considerations that we'll talk about how we design the compound to engender that particular effect. Because we're using an IgG backbone, which is an immunoglobulin backbone, that's a well-described, well-understood biological product that's typically administered in patients every 2 to 4 weeks in an outpatient setting. So we think it's very flexible for patients and reasonably convenient. And then importantly, we've had great success thus far with our partnership with Lonza, a global manufacturer of complex biologics like CTIM-76, where we are currently scaling up manufacturing of this protein to support human trials, and that relationship has gone really well. So to sum this all up, we think we have a really interesting asset that can potentially address Claudin-6 driven cancers. And so with that, I'll advance 2 slides and turn it over to our scientific lead, Dr. Eric Butz.
So Claudin-6 is the target of CTIM-76 and is associated with worse prognosis in a number of different cancers. 3 of them are shown here, and you can see that Claudin-6 high tumor cells have faster progression and worse overall prognosis than Claudin-6 low tumors. Claudin-6 proteins are a family of tight junction proteins. It's a large family with 27 different proteins having been described, and dysregulation of Claudin-6 proteins occurs in multiple diseases, including a number of different kinds of cancer. Normal function would be the red molecules in the cartoon on the right-hand side, and where they're shown to be gluing cells tightly together to form tight junctions. and limit traffic of molecules across those junctions. Claudin 6 is part of the wider Claudin family, and in particular, there are 3 other closely related Claudins: Claudin 9, Claudin 4, and Claudin 3. And one of the challenges of targeting this is that there's a high degree of similarity between Claudin 6 and the other 3 Claudins in that group. And while Claudin 6 is an oncofetal tumor antigen, the other 3 Claudins are widely expressed in normal tissues, and particularly Claudin 3 in the pancreas, Claudin 4 in the kidney and pancreas, and Claudin 9 in the ear. So specificity in targeting Claudin 6 is important to avoid off-target toxicities. In particular, we think that Claudin 6 has potential to reach patients in a number of indications with high unmet need, principally testicular cancer, ovarian cancer, and non-small cell lung cancer, where expression of Claudin 6 is quite high. With testicular cancer, it's in almost all cancers. In ovarian, it's about 60% of ovarian cancers, and in non-small cell lung carcinoma, with a pretty high unmet need, about 30% of cells of tumors express Claudin 6. And here's some staining data that backs that up. You can see on the right-hand panel, there's a wide variety of normal tissues which do not express Claudin 6. And on the left-hand side, you see examples of testicular cancer, ovarian cancer, and lung cancer, and high levels of Claudin 6 expression in those cells. Claudin 6, we think, makes a really excellent target for a bispecific antibody. It's tumor specific, at present at high density across many cancers. It's expressed at low levels or absent from normal tissue. And as Joe Rucker will be explaining here in just a moment, there's quite a bit of complexity in targeting the antigen, which we think we have a good reagent to do. And it's closely related to its nearest neighbors, but we have a high degree of specificity. And recently, BioNTech has published data from a CAR-T project showing that they had a 50% response rate in their responses to the CAR-T cell, providing biological proof of concept. But with a bispecific, it would be a much simpler dosing strategy than for a CAR-T. So we think CTIM-76 is selective for Claudin 6, potent in vitro experiments, which Joe will also be providing you some evidence for, with potential for a wide therapeutic window and excellent manufacturability because of the backbone structure.
Okay, so what I'm going to do now is talk about where CTIM-76 came from, and then talk a little bit about the end, about where it's going. So when we were first beginning this program, we began to first talk about, what arms of the Claudin 6 and the CD3, what should they look like? So in terms of the Claudin 6 targeting arm, we had developed a high affinity Claudin 6 antibody. But in particular, we got the high affinity, but we're able to maintain very, very high specificity for Claudin 6 against the closely related Claudins, Claudin 3, Claudin 4, and particularly Claudin 9, which only differs from Claudin 6 by 3 amino acids in the extracellular domains. We thought about the different CD3 engaging arms, and in terms of those arms, we picked arms that were clinically validated, where we had a freedom to operate, but we did have a range of potencies that we could choose from when we were scanning through these different possibilities. We chose a variety of formats. We didn't make any assumptions about what format we felt would work best. So we did BITE formats. We did formats where we had single arms binding Claudin 6 and CD3, arms where we had bivalent Claudin 6 and CD3 binding, and a format where we had bivalent binding to IgG, bivalent binding to Claudin 6, but only monovalent binding to CD3. And so we were able to use all of these to create a very large, comprehensive panel of bispecifics to look for the ones, bispecific that gave us the best properties. Just some other factors. Of course, we wanted cross-reactivity to non-human primates for both the arms, which would help simplify preclinical studies. We used effector function silencing just to make sure that to control immunogenicity. And, for the scaffolds, which had IgG-like folds, we maintained binding of the neonate FC receptor for half-life extension. So our initial panel generation is we took the variety of Claudin 6 arms, a variety of CD3 arms, mixed it into these variety of bispecific formats, and ended up with 54 candidate molecules. And we did extensive characterization, functionally, safety profiling, developability, and ended up eliminating, choosing 4 lead candidates, which had optimal properties in all of these 3 areas. These 3 candidates were one BiTE molecule, 2 molecules that had a 1-by-1 format, so an IgG where they had a single arm Claudin 6, single arm CD3, and the differences between those 2 were on the Claudin 6 arms where there were subtle differences in affinity and specificity. And then one final molecule, which was a 2-by-2 bivalent for Claudin 6, also bivalent by CD3. And these were arms that we then did extensive characterization to end up choosing CTIM-76 as our lead candidate. So CTIM-76, which was, once again, is a 1-by-1, we looked at this in a variety of characterization. Here I'm showing T-cell directed cell cytotoxicity assays in 2 different cell lines, a K562 line overexpressing Claudin 6, where CTIM-76 showed sub-picomolar cytotoxicity compared to a slightly weaker cytotoxicity with other molecules. We also went into an OV90 cell line as well, also showing a low picomolar potency as well. And so in these assays, CTIM-76 was either equivalent to or superior to the other formats. It's well known that bispecific molecules, CD3 engaging bispecific molecules, is CRS, cytokine release syndrome, can be a concern. So we did extensive cytokine release profiling. And what we show here is that we did a direct comparison of CTIM-76 and a BiTE molecule that had the exact same arms, this identical Claudin 6 binding and CD3 binding arms. And what you see here is we show the cytotoxicity for the CTIM-76 versus the BiTE. CTIM-76 is more potent. And we also see cytokine release for these 2 sets of molecules, for the CTIM-76 in red and the BiTE molecules in blue. And we see that we have a good separation between cytotoxicity and cytokine release in CTIM-76 that is equivalent to or superior to the separation we see for the BiTE molecules, saying that we should have a good range of doses that we can work with as we develop this molecule. But I think the biggest concern was specificity, and where we really we're trying to have the most optimal arm as we could. As mentioned, Claudin 6 only differs from Claudin 9 by 3 amino acids. And so getting that specificity for Claudin 6 versus Claudin 9 was paramount for the success of this program. So we, our molecule, the parental IgG molecule, which is IM301, which is shown on the right, shows good specificity for Claudin 6 in red versus the other Claudins, and has a rather unique epitope shown in the figure. On the left is IMAP27. This was the Claudin 6 antibody that was developed by Ganymed, later licensed by Astellas. Somewhat different epitope from IM301, from the CTIM-76 Claudin 6 binding arm. And as you can see, while there is some specificity for Claudin 6 over Claudin 9, there still is extensive binding at Claudin 9 of this molecule, at least in the IgG format. We wanted to dive down into this further, looking at the Claudin 6 binding in the CTIM-76 molecule itself. So what you see here on the left is the binding of CTIM76 to Claudin 6 expressing cells, and it has a really strong nanomolar, slightly subnanomolar binding to Claudin 6. On the right is the binding that we see to Claudin 9, and as you can see, up to 1 micromolar, we see no binding to Claudin 9 for CTIM76, giving us several orders of magnitude of specificity. But binding wasn't sufficient for us, and we know that particularly in T cell engagers, the cytotoxicity can be very sensitive to low levels of off-target binding, and so we didn't want to just trust the specificity that we saw in binding. We wanted to go on to cytotoxicity assays to really dig down into this. So what you see here are cytotoxicity assays on cells that are expressing Claudin 6, Claudin 9, Claudin 4, and Claudin 3. Claudin 6 by cytotoxicity, as we mentioned before, is in the sub-picomolar range, so very, very potent. Claudin 9, Claudin 4, and Claudin 3, although we saw in the binding in our binding assays, actually do show some level of cytotoxicity. And while this is 1,000-fold, roughly 3 orders of magnitude difference in the cytotox that we see for Claudin 6, there are doses where you will see cytotoxicity against these other Claudins. And this really is a good take-home message, which is binding can show you specificity, but functional assays can be much more sensitive. And even though you may not see binding to these off targets, it's only by testing in cytotoxicity assays that you can really convince yourself that the specificity that you're seeing is what you need. So let me just tell you sort of summarize this data and tell you where the next steps are. We're seeing encouraging efficacy signals where xenograft studies are ongoing right now, but we're seeing encouraging signals, and we're seeing selectivity across both binding and cytotoxicity in vitro, as I showed you in the previous slide. The tolerability studies, the safety studies are beginning, so we're seeing no significant safety findings to date. As we showed in the cytokine release assays, we see preferential cytotoxicity over activation of cytokines, telling us that we should be able to find doses which have that good cytotoxicity and minimize any safety concerns. And the PK is consistent with an IgG backbone. Once again, one of the advantages of the CTIM76 format. And we're making good progress towards the clinic. So IND enabling studies are ongoing, and the IND submission is expected in Q1 of 2024. And at this, I'll hand back to Marty Witt.
So when we think about how one should view Claudin 6 in the context of there being a lot of interesting cancer targets out there, there, unfortunately, in the world are a lot of different cancers, and we view there's a fundamental unmet need in almost all of them. But there's a lot of opportunities, and AACR is a great example where many companies are presenting many different ideas. And the lens that we typically think about for interesting cancer targets is through 2 matrices. So how big is the total addressable market, which is the horizontal axis, and then the vertical axis would be how validated is that target. And so sometimes there's very new science where we don't know a whole lot about what's going on with the biology. That would be an emerging target. And then on the far end, there are validated targets, things where there are clinically approved drugs. In the case of Claudin 6, our competitor BioNTech has publicly reported on 21 patients worth of data in testicular and ovarian cancer showing very, very promising efficacy, where they're shrinking tumors in about 50% of the patients. And if you look at the total addressable market, as my colleague Eric showed earlier on, we think there's directionally 62,000 or more patients in the United States alone who have Claudin 6 positive cancers. So there's a really large opportunity for this novel, interesting target. As we think about how one might therapeutically exploit Claudin 6, we think really bispecifics and CAR T, so the immunotherapies, are the strategies there are likely to yield the highest success. In the case of monoclonal antibodies, Claudin-6 does not have a ton of signaling activity. And so a monoclonal would have limited activity, and that's been shown clinically by Astellas. Secondarily, we also do not believe that antibody drug conjugates would make for good Claudin-6 therapeutics. And really there's three reasons for that. So first, we disclosed in December at an R&D webinar that one of the key criteria for antibody drug conjugate success is to get the antibody drug conjugate into the cell of the tumor, which is referred to as internalization. We do not see Claudin-6 as being a protein that internalizes, at least in our hands. Secondarily, we know that Claudin-6 functions to enable cells to attach to one another. And there's a lot of Claudin-6 expressed per cell in cancer cells, about 500,000 copies. And so with this high Claudin-6 expression, the cancer cells bind really tightly to one another, forming this almost impenetrable ball. That makes it very hard for drugs to get deep into the tumor. And so one of the ways the antibody drug conjugates work is by binding the surface, not actually internalizing, but just releasing drug near the tumor, hoping that it gets in. And we think that's unlikely to be successful given that these tumors are chemo resistant and bound so tightly. And then the last thing that gives us reservations about ADCs is the fact that a company called Stemcentrx put an ADC in the clinic and it had very substantial toxicity due to off-target binding to Claudin-9. And this is a good transition slide. As we think about our competitors and their selectivity profiles for Claudin-6, they actually look quite similar to Stemcentrx. All that being said, a group from UCLA formed a company called TORL, T-O-R-L Biotherapeutics, that announced a $158 million round on Friday. So, and that round was led by Goldman Sachs with participation from well-known funds such as Perceptive and Deep Track Capital. And then actually Bristol Myers Squibb participated in that financing. So it's a great day for Claudin-6 when a major pharmaceutical in BMS and several of the top biotech specialist funds are betting on Claudin-6 as a therapeutic modality. So we really think that bispecifics, though, afford the best approach in this area, and we really view it as having a limited competition set therein. We believe that Amgen is our primary competitor, at least as it relates to selectivity, and they've only disclosed selectivity versus Claudin-9. And as my colleagues mentioned earlier, Claudins 3, 4, and 9 are all important off targets for Claudin-6 bispecifics and therapeutics in general. And so we think we are differentiated based on selectivity. And we are very interested to see Amgen as they disclose more data about their selectivity profile going forward. So it's still very early days. And we get questions a lot of how far behind are you from other people. Amgen just opened their trial last month. BioNTech has dosed 21 patients. So it's still early days. I think we absolutely have an opportunity to catch up to the major players here and start generating additional data. So slide 26 just is a summary of what I outlined. And so to take it home, the field is emerging. We think Claudin-6 is a really novel, important target that one should consider for solid tumors. And the TORL financing, raising $158 million from well-respected biotech specialist funds, as well as Bristol Myers, I think further helps provide external validation for the target. So with that, I'll pause here and I'm going to turn the dialogue over to my colleague, Sue Romanoff from our investor relations firm, Edison, to guide a discussion. We got a number of questions during the conversation. And so maybe we'll go one by one and have my colleagues, Joe and Eric, address them. Thank you.
Great, thank you. As we mentioned earlier, if you have any questions, please enter them through the Q&A portal at the bottom of the Zoom panel. We'll just go through a few of these here right now. What defines high and low Claudin expression, and how is this validated?
I can handle that one. So different publications have looked at that in a number of different ways. It's been looked at by RNA and by IHC. We think that RNA expression parallels pretty closely to protein expression. And the work that we've done has used a number of different cell lines where we think that the number of antibody binding sites ranges from about 500,000 at the high end to something like 50,000 at the low end. And we see good CTL activity in that whole range with different EC50s, depending on how many targets there are per cell.
Here's another one. Given the phenotype similarities of Claudin-6 with other Claudin molecules,
So
is
you
there any signals to suggest risks, evidence of off-tumor organ toxicities? Also, what kind of risks of cytokine release syndrome based on cytokine profiling?
So we've looked at where we start to see in vitro. We've looked to see where we see cytokine production as opposed to killing. And in the in vitro assays that we've done, we see about a 2-log shift in the cytokine production versus the killing. So we think we have a pretty big window in terms of where we should see effective target killing and where we would begin to see significant release of cytokines in vivo. And then in terms of off-tumor toxicities, we think that they should be minimal with Claudin-6, where we have good specificity. The BioNTech CAR T cell approach showed a little bit of toxicity, but nothing too serious. And a lot of that was liver toxicity. And we suspect that some of that may be due to the vaccine boost that they were doing, and the nanoparticles. Many of them will be caught up in the liver, so you'll get liver expression of Claudin-6, which would render that system susceptible to some liver damage.
For testicular cancer, it has a high expression of Claudin-6, even though in smaller end market, it is a smaller end market. How feasible is it to dominate the indication with CTIM-76 given other therapies available?
Yep, I can handle that one. So testicular is a great example where by and large, it's caught early. 95% of the patients will be cured via surgery. And so the real challenge is when it comes back. And at that point, there are very few treatment options. If you look at historical response rates, meaning the percent of patients who see their tumor shrink with a particular therapy, testicular, you're looking at the high end, maybe 15 to 20% of patients having a response. And what BioNTech showed was 50%. Now, there are CAR T therapy, that's a $2 million treatment. And so convincing an insurer to pay for something like that is a steep hill to climb. In our case, we really do believe bispecifics can mirror that activity, but do it in a manner that is much, much more cost effective for insurers and much easier for patients. So there's a variety of strategies in drug development. Some companies go after the largest indications, the largest market size. Those are typically the most competitive, most expensive ways to go about. Some view a beachhead strategy. So you pick a very small cancer like testicular, you try to go for a quick approval, and then you build out from there. We have not disclosed what our development strategy will be, but we have disclosed that testicular, ovarian, and non-small cell lung are at the top of our list right now.
Great. Maybe Joe, were other cytokines aside from TNF and INF evaluated, since it's likely that other cytokine drivers drives CRS, IL-6, IL-7, etc.?
Yeah, it's a great question. I mean, we did test other cytokines. For example, we looked at IL-6, IL-2, and others, and the profiles we saw with those cytokines were quite similar to the profiles we saw with TNF alpha and interferon gamma. As we move this project forward, of course, we can expand into other cytokines. I saw some interesting posters at AACR, for example, IL-8 with regards to T-cell engagers. So I think, as we move forward, we can expand to additional cytokines, but we have looked at IL-6, for example.
What is the frequency of antigen escape of Claudin 9 after antigen pressure from CARs or bites?
Given the depth of clinical experience right now, it's impossible to say. So BioNTech's had 21 people on their bite so far without long-range follow-up. Amgen's only just started their clinical trial, so that remains to be seen.
Maybe Martin, why not partner with Amgen and pool resources?
If Amgen shows up with a big check, that seems very reasonable. But pithy comments aside, the fundamental challenge we have in front of us is BioNTech and Amgen are very substantial competitors, not just from a capital-based standpoint, but from a resource standpoint. And so one of the things that we're evaluating as a company is we've guided that we have capital in the late 2024. Maybe we can extend that a little bit beyond that through creative means, but to really compete with the larger companies in a substantial way, we'll either need incremental capital, a partner, or potentially sell the asset. And so all those paths are being evaluated and will continue to be so in the future.
Okay, great. I mean, if anybody else has any other questions, otherwise we can wrap this up, Martin.
There was one additional question: is testicular an orphan indication? And it is an orphan indication. And so for those of you unfamiliar with orphan, that's a potential regulatory designation to have increased regulatory interactions with the FDA, a potential accelerated path toward drug approval, and then a company, should the drug get approved, is eligible for R&D tax credits that are retroactive.
That's all the questions that we have time for today. Thank you for taking time to join. If you have any additional questions or would you like to speak to the management team, don't hesitate to reach out to the contact information on the Context website. Thank you.